Hello everyone, and welcome to the end of our discussion on muscle tissue. So in lecture, we talked about the anatomy of skeletal muscle tissue, properties of muscle tissue in general, and we ended talking about a region called the neuromuscular junction. So we should have everything that we need to talk about how a skeletal muscle contracts. That's going to be our major focus for today's lecture, followed by some clinical connections, talking about some disorders, and then we'll end talking about cardiac, smooth muscle tissue, and then a little bit about development. Now, recall from lecture where the neuron meets the sarcolemma. This is called the neuromuscular junction, right? Our NMJ. And the axon is going to release neurotransmitters, little dots, that will bind to receptors that are on this structure here called the motor end plate. So remember, what were these neurotransmitter called, right? This was acetylcholine, ACH. Now, we're not going to get into as much detail as we will in physiology, but this is just a really great overview of how we get that connection from our frontal lobe to move our fingers, to how that actually happens. So what's going to occur is, obviously, first and foremost, we have a signal from the brain. This is a voluntary contraction we're talking about. So what occurs is we get a signal through a neuron. It's going to go down this motor neuron and end at the neuromuscular junction. We're going to release the acetylcholine. It's going to bind to receptors on the motor end plate, and that's going to create the action potential, the electrical signal that will propagate down the sarcolemma, down the T tubules, and then open up some voltage-gated channels. So we're going to kind of talk about how we connect this electrical signal to the actual mechanical action of contracting a muscle. Now, the first thing that we're going to talk about, and this is going to sound a little backwards, I like to describe the way this is taught as sort of Tarantinoing it. So if you think about a Quentin Tarantino movie, right, they kind of show you what happens at the end, or at the beginning, and then they skip to the end and don't really tell you about the middle until later on. That's kind of what we're doing. I just showed you the beginning with the action potential getting formed from the acetylcholine binding to receptors on the motor end plate. And now we're at the end. We're looking at the end result, which is the contraction cycle, where the actin and myosin are going to slide over each other. And then we'll do a flashback and look at what happened in between. So the contraction cycle of myosin and actin is a four-step process. So the first thing that happens is cocking. And this typically has already occurred prior to any inclination that you're going to contract the muscle. Actin and myosin really want to be together. But remember, we have troponin and tropomyosin that are not allowing that to happen. There are regulatory proteins who are going to prevent unnecessary connections happening. But because myosin really wants to be with actin, it's going to be ready so that if the opportunity arises, they can connect. So during the process of cocking, we take an ATP, remember that is our energy currency, and we're going to break it down. And that process of breaking ATP into its constituent parts, ADP and a free phosphate, is called hydrolysis. And you guys will learn a little bit more about this in physiology. It's basically using water to break a bond. So we have this energy stored up, ready to go. Once the signal has traveled down all the requisite parts of the skeletal muscle and the actin binding sites are available now, the myosin head will reach up and grab actin. And we call that a cross bridge because if you think about it as these being individual bridges, when you connect two bridges together, it's called a cross bridge. So now they're physically connected. The next thing that happens is we're going to perform a power stroke. So what happens with the power stroke is I've grabbed onto actin here and I'm going to move it like this. I'm going to bend it towards midline. How does this happen? One thing you want to remember and take this with you into physiology is whenever something binds to or unbinds from a protein, it's going to change the protein's shape. So when we want to move actin towards midline, what you'll notice is that ADP and free phosphate leave. They leave the myosin head, so something has left a protein. That means it's going to change shape. So now instead of being like this, it bends forward, okay? Pulling actin towards midline. What's really important to understand about this contraction cycle is it's not a 3, 2, 1, everybody heave ho. It's more like a ratchet or like when you're using a screwdriver. So you have to do it multiple times before it's unscrewed or screwed in. So we're ratcheting the actin towards midline. So that means we need to do this again. And the final stage of the contraction cycle is going to be relaxation or releasing. So what's going to happen is myosin and actin are now bound, but I need to reset so they can reach up and keep sort of doing this weird ratchet. The only way that myosin will release from actin is by having another ATP come in and bind it. So a new energy molecule comes in, something has bound to a protein, so it's going to release from actin. It's going to hydrolyze that ATP, reach up, form a cross bridge, ADP and free phosphate leave, we get a power stroke, and this continues until we get all the way to midline. We can no longer contract anymore, or the signal stops. So this is the actual shortening of the muscle. So how do we regulate this? How do I make sure that that contraction cycle happens when I want it to happen? That's where the regulatory proteins come in, troponin and tropomyosin. So remember, tropomyosin is going to be the little thread, okay, that's blocking myosin. So that's a really good way to remember it. Tropomyosin blocks myosin. Troponin is going to sit in groups of 3 on the tropomyosin string, okay? And there are 3 different types of troponin. You'll learn more about this in physiology. But for us, we're looking at one of the troponin molecules here that's going to bind to calcium. So when we have no calcium, as you can see from this sectional view, the tropomyosin and troponin are blocking those little blue dots, which are the actin binding sites. Now, if we look at this sectional view, now we've added calcium. It's bound to troponin. Once again, when something binds to a protein or unbinds from it, it's going to change its shape. So when calcium binds, it's going to change the shape of the troponin-tropomyosin complex, and now myosin, which is the red here, can actually bind to actin. So the thing we're getting at here is the way we regulate the contraction is by the concentration of calcium. If I add calcium to the system, it's going to bind to troponin and move tropomyosin, and then the whole contraction cycle can now start. So where does the calcium come from? Well, if you recall from lecture, we have an organelle in our skeletal muscle called the sarcoplasmic reticulum, and that is where calcium is stored. So if we look at a very poor drawing, I'm going to kind of draw a T-tubule here. So here's my T-tubule. Switch our colors a little bit. And then this green blob that's a butt to it, the webbing, this is the SR. And if you recall from the previous lecture, there are little proteins that sit where the T-tubule meets the terminal cistern. These are voltage-gated calcium channels. And what do these do? When that electrical impulse moves down the T-tubule, it's going to lead to a change in membrane potential. So it's going to electrify the membrane. Those voltage-gated calcium channels are always closed because if they're open, the calcium just leaves through the process of diffusion. So they only open when there's a change in voltage, hence the name voltage-gated. When the electrical impulse runs down, it's going to change the membrane potential and it's going to cause the doors of those channels to open. And that's going to cause the release of calcium into the sarcoplasm, which is then going to bind to troponin, move tropomyosin, and allow the contraction cycle to begin. So this idea where we take an electrical signal from a neuron and we turn it into a mechanical action like flexion, okay, that is called excitation-contraction coupling. We are coupling excitation, the action potential, to the shortening of muscle. And calcium is the driving force. I need the calcium so that I can get the connection between actin and myosin. And when there's no calcium, there's no connection, there's no shortening. So when we have a contraction, just in very, very simplistic terms, because once again, we're not in physiology. In very simplistic terms, the calcium gets released because of that change in voltage from the action potential, binds to troponin, moves tropomyosin, and then when we want to relax the muscle, we kind of reverse everything. So we want to get rid of that calcium. You're going to pump the calcium back to its storage site here in the SR. And as those calcium levels drop, the troponin and tropomyosin complex comes back together over the actin binding sites, and we can no longer have a connection between actin and myosin. So if you guys want to pull up your PDF, I can't really go back and forth when I'm recording, and have an image of that myofibril with all of the structures or the neuromuscular junction, I'm going to kind of walk you through what's going on. So first things first, once again, we get a signal from our brain. It's going to travel down a neuron, in a nerve. It's going to end at the axon terminal. That is going to release acetylcholine into that synaptic cleft. It's going to bind to our receptors on the motor end plate, which is, remember, is a specialized region of the sarcolemma. That is going to in turn create an action potential, the electrical signal that travels down the sarcolemma and then down the T-tubule. As it goes down the T-tubule, it opens voltage-gated calcium channels who release calcium. That calcium is going to bind to troponin, which will move tropomyosin. And now the actin binding sites are available. Myosin, which is already hydrolyzed in ATP, can now reach up, grab actin, forming a cross bridge. The ADP and free phosphate from that hydrolysis is going to leave the myosin head, and it's going to perform a power stroke as it ratchets one click over the actin molecule towards midline. Now we release from actin as a new ATP binds to the myosin head, and we go through the cocking, cross bridge formation, power stroke, and release phase until we either make it all the way to midline or we stop sending a signal. When we want to stop that contraction, we basically stop the signal from the brain, we remove the acetylcholine from the synapse, we gather all of our calcium back into the sarcoplasmic reticulum, and everything just kind of goes back to its original shape. Okay, so that's basically what's going on every time we contract a muscle. Now, once again, at the beginning of this chapter, there is a really great video that runs through this whole process that I highly encourage you all to watch. And if that's not the video that's linked there, it's definitely on the Wikipedia page. Now let's kind of talk about some disorders that are associated with some of the parts or processes that we've looked at in regard to skeletal muscle. So the first thing I want to look at is myasthenia gravis. Myasthenia gravis is an autoimmune disorder. So autoimmune, auto means self. It's where your immune system attacks your own body tissues. We still aren't 100% sure why these autoimmune diseases actually occur or why they're triggered. But in the case of myasthenia gravis, what's going on is the antibodies, the immune system, is attacking those receptors that are on your motor end plate. So as you can see, as we reduce the number of available receptors, that means I'm going to reduce the ability to actually sort of turn on my muscle. So what happens is you're not getting enough of a stimulus to trigger a strong enough action potential to get that muscle to work. So you typically see things like weakness and fatigue in facial muscles. It can affect the eye muscles. Eyelid drooping is a very common sign of myasthenia gravis. And how it's treated is by using what are called anticholinesterase drugs. So there is an enzyme that lives in the synapse called acetylcholinesterase. You don't need to worry about that particular enzyme. You will in phys. What acetylcholinesterase does is when we're ready to stop a muscle contraction, it basically eats up the acetylcholine. Now, if I don't have enough receptors, I want to try to keep the acetylcholine in that synaptic cleft as long as I can, because the more acetylcholine I have, the likelihood that I'll actually get a strong enough contraction will increase. So when you take these anticholinesterase drugs, it reduces that enzyme and promotes the longevity of the acetylcholine. So that's kind of how we treat it. There's currently no cure, but that's sort of the treatment that we have right now for myasthenia gravis. So once again, the immune system is attacking the receptors on the muscle itself. Now, Botox is not a disorder. Botox is a medicinal compound that can be used for a bunch of different reasons, both cosmetically as well as for treatment of different disorders. So Botox is a form, it gets its name from botulism toxin, from Clostridium botulinum, which is a very smelly anaerobic bacterium. And what botulism toxin does is it's going to block the release of acetylcholine. So if you get botulism from improperly canned goods, it can actually lead to death, because if it attacks the nerves at your diaphragm, they won't release the acetylcholine and your diaphragm won't get the signal to contract. So it can be deadly. Botox injections are basically purified forms of this toxin that are injected strategically into different places. So if we think about the cosmetic use of Botox, it's going to be used to remove wrinkles of the forehead, in between the eyebrows, and around the mouth. What happens is when we inject that Botox, a lot of times the wrinkles here in the forehead are because, and in between the eyebrows, are because of mild contractions of those muscles. We have our corrugator supercilii here, that's your like frowny muscle. And so if we can paralyze those muscles, then the wrinkles will go away. So it's not a filler, it's a paralytic that's going to cause the muscles that are causing that wrinkling to happen to relax. So that's why individuals that have had Botox, they can't make certain facial expressions, lifting the eyebrows, smiling, things like that, because you've paralyzed the muscle temporarily. Now, as I mentioned, it's also used for actual treatment of disorders. So one of the treatments it's used for is for something called hyperhidrosis. So hyper, let me write this out, hyperhidrosis, hyper means a lot. I think I spelled that wrong, hydrosis. Hydrosis meaning like liquid water. So it's excessive sweating. Like, not like, wow, I'm sweating right now so much, but like your hands are consistently wet, your armpits are consistently wet. This is because in order to sweat, we need to get a chemical signal from a neuron. And when we stop that chemical signal, the release of acetylcholine on that sweat gland, we're not going to have the sweating. So you can have targeted injections in, the wrist or the hands or the armpit to help reduce that sweating. So that's one of the ways Botox can be used therapeutically. Now in this class, we don't really get into a lot of detail about cardiac and smooth muscle tissue like we just did with skeletal muscle. That's because cardiac muscle tissue and smooth muscle tissue are very, very complicated in terms of their physiology. We add in additional compounds. It gets a little confusing, especially with smooth muscle. Smooth muscle is just very, very weird. So we're going to save that for physiology. What I do want to talk about is just, once again, reiterating some differences between cardiac and smooth muscle tissue and skeletal muscle. So remember, cardiac muscle tissue, this is autorhythmic, just like some of our smooth muscle can be. Remember, auto meaning self, so it can set its own rhythm. It can contract on its own. It does not necessarily need a neuron to tell it to contract. Now your heart does have that. We'll talk more about that as we get into the heart lecture in a couple of chapters from now. But the heart sets its own rhythm, and it does utilize extrinsic nervous stimulation to change its heart rate and things like that, but for the most part, it sets its own rhythm. The contractions are longer, and the action potentials are longer. And as you'll learn in physiology, that's because one of the things that can happen in skeletal muscle is something called tetany, where the muscle just stays contracted for a long time, like tetanus. That's why you get a tetanus shot, because you don't want lockjaw and things like that, where your muscles are just chronically tightened. The length of the action potential in cardiac muscle makes it so we can't fire another action potential and lead to that state of tetany. Because if your heart muscle goes into tetany and stops contracting, you're going to stop circulating blood and basically cardiac arrest. As we saw in the histology, the actin and myosin are arranged in the same patterns. That's why we see those striations in cardiac muscle tissue. We have those dark vertical bands that are the intercalated discs that are going to allow a very fast movement of ions like sodium, so that we can get a very uniform contraction throughout the heart. As you are probably aware, the cardiac muscle tissue requires a high amount of oxygen. So it's aerobic, uses lots of oxygen, and it has a lot of mitochondria. Way more than skeletal muscle, because skeletal muscle typically gets a chance to relax, replenish its stores. Cardiac muscle doesn't get that. There's about 0.8 seconds when your heart has this period of relaxation, and then it just starts contracting again. So that's your diastolic period. So we have to make sure that there's enough mitochondria inside the muscle cells that they can continually regenerate their ATP needed for that contraction cycle. Now, smooth muscle, once again, smooth muscle is really funky. It has actin and myosin. It is involuntary, like our heart muscle. Some of them in the, for example, gastrointestinal tract are autorhythmic. So we do have populations of cells that trigger action potentials without outside nervous stimulation to move things along in your GI tract. But for the most part, it's primarily just involuntary in the fact that we don't think about sending that signal. So when your pupils change size in your eyes, you're not telling the smooth muscle in your eyeball to dilate or constrict. It just happens. So remember, the easiest way to remember where smooth muscle is found is just to think of the walls of hollow organs. So that means the GI tract, blood vessels, your airways that go to your lungs. But we also have them in the erector pili muscles. Remember, you guys saw that in the video on the integumentary system. It's the small muscle that's attached to a hair that will allow it to stand up on end. And then, of course, as I mentioned, we have muscles that change the shape of your pupil from really small to really big, typically in relation to light. And then we also have changes in the shape of the lens, which is this sort of magnifier inside the eyeball. And we'll talk more about how that works at the end of the semester. So when you look at smooth muscle, remember, when you look at it under the microscope, it's smooth. It's these spindle-shaped cells. We don't see striations on it. But that's not because there's no actin or myosin. It's just simply because the actin and myosin are arranged in a different pattern. So they're not stacked on top of each other where we get these dark and light bands. They're kind of arranged in an oblique sense, so at an angle. And there's no Z discs. So when we look at the sarcomere, we're going to see that we do have thick filaments and thin filaments, and the thin filaments, the actin, is attached to some sort of connecting point, like it is in skeletal and cardiac muscle, right? Those are the Z discs. But in smooth muscle, there is no Z disc. We call this a dense body. So all these little dots are dense bodies that are connected through intermediate filaments. And when we go through the contraction process, which once again is very weird, the actin will slide over myosin, just like we talked about, but it does it in a different fashion. And so when they contract, it kind of creates this, like, looks like a fat worm. So we're still getting shortening, but we don't really have a typical sarcomere, and they're not arranged in the typical manner that you would think of when you think of cardiac and cardiac and skeletal muscle. So once again, the whole process you guys will learn about in physiology. Now, the last thing to talk about today is, where does muscle come from? Now, remember, we have 3 types of germ layers or germ tissue, embryonic tissue. We have our blue ectoderm, our red mesoderm, and our yellow endoderm. So when we're looking at where muscle is coming from, it's derived from mesoderm. Now, what we're looking at here is an embryo that's almost a month old. Okay, so 22 days. This is your neural tubes. This is where your nervous system will be sort of created. Talk more about that when we get to development. And on either side of it, we see what are called somites. Soma means body. So we have a sclerotome, a myotome, and a dermatome. What we're concerned with is this middle somite called the myotome. Myo, remember, means muscle. So the myotome is what's going to form your skeletal muscle. Where do the other types of muscle come from? There's a lot of other mesodermal cells that are found throughout the developing embryo. The way development occurs is a lot of chemical signals telling cells to move different places and to differentiate. So some mesodermal cells will be told to become smooth muscle. Others will be told to become cardiac muscle. So cardiac and smooth come from mesodermal cells, and skeletal muscle is going to come from the myotome. Okay, so that is where everything comes from. Okay, and that is it for muscle tissue. In our next lecture, we will talk about the introduction to muscles themselves, how they move, how they work together, and start talking about the head, neck, and trunk. And I'll see you guys there.